Click for IB Biology. We're looking at the organelles known as the mitochondria and the chloroplast. There's a blank PDF of this presentation down in the description with the usual 10 questions with their answers so that you can check your progress. So let's take a look first at the mitochondria. So a mitochondria, when seen in a micrograph, could be anywhere between 1 to 10 micrometers long, and their number of mitochondria within a cell is often a reflection of the demand for ATP within that cell. So of course, the function of the mitochondria is the synthesis of ATP, and we know that this takes place in the process called aerobic respiration. Now, this video is not concerned with the details of aerobic respiration. We have those videos that you can go and look at. What we're actually concerned with here is recognizing the adaptations in the shape and form of the mitochondria which support that process. So of course, this is one of the reoccurring ideas, is that shape tells us about the function, and the idea is that the specialization of the shape enhances the function and allows it to occur efficiently, or we might say adaptation. And so the mitochondria, when seen in a micrograph, has around the outside has double membranes. And of course, as you'll know, this reflects the endosymbiotic theory which we have looked at elsewhere on the course. So there's a double membrane. Now the inner membrane actually folds inwards like so. The outer membrane is consistent like so. And these folds in are called cristae. And so we're creating a number of different spaces within the mitochondria. There's the inner matrix, which is the location of the Krebs cycle. That's an enzyme cyclic metabolic pathway. The cristae membranes create this space here, and that space is used to concentrate hydrogen ions in the synthesis of ATP. So if you look up the videos on that, especially what's called the electron transfer system, you'll find that the space created by the cristae is important as a way to concentrate protons to drive the enzymes which generate ATP. So the folding increases surface area, of course, for the enzymes and the electron carrier system. Now, the interesting thing about the cristae is that in recent times it's been discovered that cristae membranes are not static, but in fact dynamic, and they form and then break down and then they reform in response to the metabolic demand of the cell, which is an interesting additional feature to the mitochondria, often not recorded in a typical textbook. Okay, so because of the endosymbiont theory, we know that there's DNA within the mitochondria. It's actually forming a loop very much like a prokaryote. There are no proteins on that DNA, again, like a prokaryotic cell. And so there's mitochondrial DNA, mtDNA, and they will produce RNA, messenger RNA, which then locates onto the ribosomes. Again, the mitochondria has its own ribosomes, 55S, again reflecting more evidence of endosymbiont theory, the very bacterial-like ribosomes. And through the synthesis of various enzyme systems, proteins and enzymes and so forth, then we end up getting the synthesis of ATP. And famously, of course, we know that mitochondria consume oxygen in the process of aerobic respiration, as is detailed elsewhere. And that actually goes right inside the mitochondria and onto the cristae membrane, where it picks up hydrogen ions to form metabolic water. So the oxygen you're breathing in now is used to form metabolic water. So the key features of the mitochondria are the folding of the membranes, the creation of spaces for localized chemistry of the Krebs cycle, and of hydrogen ion concentrations, folding of the membranes to create surface area for enzyme systems. Let's take a quick look then at the chloroplast. Again, we're looking at the chloroplast from a structural adaptational point of view. Now, the chloroplast, of course, is the organelle associated with photosynthesis. And in photosynthesis, what we have is the conversion of light energy into chemical energy. So let's take a look at the adaptations which promote this transformation of energy from light energy into chemical energy. And we start with our micrograph. Again, much like the mitochondria, we discovered that the outer membrane of the chloroplast is again double, but in this case, as you can see from my sketch diagram here, there is no folding of that outer membrane. So that's endosymbiont theory once more. Within the chloroplast, there are stacks of membranes which are called thylakoid membranes. And the significance of those, as you can see from my diagram, is that this is the location of the molecule called chlorophyll, the pigment called chlorophyll. And of course, what chlorophyll does is absorbs the light energy and turns the light energy into chemical energy, initially as electrons, but ultimately in the conversion of the energy from light into electrons, the chemical energy, it will end up in the form of ATP again, and something called NADPH, the details of which are in the videos on photosynthesis. And as a waste product, again from the thylakoid membranes, we have oxygen, which is derived from a process which is called photolysis. Spell photolysis, but pronounced photolysis. Now, once the energy has been converted into chemical energy of ATP and NADPH, then the chloroplast has also created this space here, which is called the stroma. And in the stroma, we have the cyclic metabolic pathway of the Calvin cycle. So the chloroplast creates this space in which the Calvin cycle enzymes can operate in isolation from the rest of the cell chemistry. And enzymes are produced in this cycle, and of course those are proteins, the genes for which are on the chloroplast cpDNA. So chloroplasts have their own DNA as well. And again, this is in a loop type structure like a bacteria. So the genes are expressed, the enzymes are made, and carbon dioxide is fixed in the formation of carbohydrates, famously, I guess, as C6H12O6. You'll recognize the structure of glucose. And so just to re-emphasize that the chloroplast is creating this isolated zone of photosynthesis chemistry. We have a light-catching region of the thylakoid membranes up here, which turn the light energy into chemical energy. And then we have a carbon fixation chemistry zone in the stroma, isolated from the rest of the chemistry of the cell. Now, sometimes we don't make carbohydrate. What we can actually do is turn this, instead we can actually make oils, and so we see oil droplets inside mitochondria, inside chloroplasts. And sometimes the glucose is joined together into long chains to form starch granules. And if we go back to the micrograph, we can see the strong representation of the starch grains here in the chloroplast. So isolation within double membranes of the chemistry of photosynthesis, the light-catching thylakoids, and the carbon-fixating stroma with its Calvin cycle, the adaptations of the chloroplast. Links to the next organelles are now being shown in the links here, link to the channel, and of course the playlist.